So welcome my dear students. So in the course of molecular biology BBT 401 I was taking the module especially 2 first and 2. In the 2 module 2 there are the DNA damage and repair mechanisms already I have discussed. There is a one thing remaining is the homologous recombination and non-homologous recombination. The important DNA damage and the repair mechanism already taught in the class and in the lecture video series in the previous is important methyl mismatch, base excision, nucleotide excision, and even SOS repair mechanism also is very important for the exam. And as in the syllabus, there is also this topic homologous recombination and non-homologous recombination. I'm just going to discuss about the things. So it is not so complicated. It's a very easy thing. So if you can remember the meiosis cell division, where the in the prophase one, you can see the homologous chromosomes, sister chromatid, they lies between each other and they exchange their genetic material called crossing over. This is the actual scenario and actual phenomenon. The similar thing is happen here. That is only with in case of meiosis cell division is with the chromosome and chromatid here or with the DNA strands. You may consider one strand like one the chromatid. Okay, so it will be more easy for you to understand. It's nothing but I am not going to study about the detailed features and all the things, but few things you need to know is that is very accurately correct double-stranded break. So whenever homologous recombination means homologous means the sequence or the strands are same, and non-homologous means not the same. Same in the term of during replication when the new strands are synthesized, just copy the existing of the or the mother copy. Okay, it's the new strand, but you can see these are the actual step. You can note it down in your notebook. This is the first number 1, 2, 3, 4, 5, 6, 7. This is the overall 7 step. And if you are able to draw this picture and you can write the full heads of the step, so you'll get full marks. So I tell you one thing, this is not so important for the exam, but it is in the syllabus. That's why I'm teaching here, rather I am discussing. Okay, so thing is that first is the homologous recombination is the homologous sequence. You can see is the same sequence is the homologous sequence. Okay, because this is the two sequence daughter duplex DNA molecule. Duplex means is the first template is the second template, two template. That's why this is the duplex. Okay, so next nucleus digests the five ends of the broken strand. So homologous recombination during the broken strand is the upper strand here, this one. So nuclease enzyme just digest at the five end of the broken strand. So it will digest here and will make the break. You can see. Okay. So it's correct. 5 prime to 3 prime, 5 prime to 3 prime. Okay. Then strand exchange by complementary base pairing. See here, this is the thing. So it's like the crossing over. You can remember the meiosis cell division prophase 1 pachytene substrate, the crossing over. So here, the broken strand exchange the base pairing with the duplex other molecule, the red one. Fine. Then once the exchange is done, then repair the polymerase synthesis DNA green part. See here and here using undamaged DNA as template. So here when it is crossed over at the end of 3 prime, this end, you can see this 3 prime is attached here. At the 3 prime end, their DNA polymerase binds, and DNA polymerase, once DNA polymerase binds, then the invading strand release broken double helix and reform. So once DNA polymerase binds, they will make them release and will start a DNA synthesis, as DNA polymerase is also there. So once DNA polymerase is there, so DNA synthesis can be started, and then followed by DNA ligation. Okay. So finally, this damaged portion, see in the green color, you can see, is completely repaired. This is the overall thing of homologous recombination. There is no complex and very tough thing. Just remember, this is the homologous nucleotide base or strand, and when the upper homologous is a duplex, because this is one set, two sets, that's why it is called duplex. And one, this is, here is the break, means the damaged portion is there. So once damaged portion is there, nuclease enzyme actually digests at the 5 prime this portion, and it will cleaved or complementary or like crossed over the another homologous base pair, then and another homologous base pair. From this another homologous base pair, the complementary strand will collect the DNA polymerase which is attached here, and ultimately once the DNA polymerase get or receive, they will make them free, and then followed by ligase. The DNA synthesis will be there, and finally ligation. So that's why at the green portion you can see homologous recombination is occur. So in this picture also the same thing you can see here. It's a little bit at the next step, means when the brakes are at the two position. Here I have discussed only one position, and here is the two position, but the thing is the same. See here at the green portion is crossing over, then crossing over, then the green portion DNA ligase, then they will make free, they then ligase product will be synthesized. This is the same thing, or maybe 2 point or 3 point or 4 point even. Okay, is the homologous recombination is the interesting thing is that this is a very interesting thing. So if you consider this is a chromosome, and this is a centromere, and here this portion, site of gene conversion, site of crossover here. Suppose, see, this is the gene, and the blue color is the heteroduplex, and here is the red color is the heteroduplex. So how the things happens? Because due to the crossing over, site of crossover, this portion at the sister chromatid is hidden here. Suppose here is the sister chromatid is crossed with that portion. So ultimately this gene is converted and relocated its position, is the conversion, and thus the heteroduplex, the blue color will be reflected here as the red color. This is not included in the homologous recombination. It's a, it's an another part actually. Okay, so you just remember these things for homologous recombination. These things, these are the points, and this is the picture. That's it. That is the enough thing for homologous recombination. And gene conversion is already discussed, and this is the non-homologous end joining. Non-homologous end joining is with the, not the duplex, it's the single nucleotide bases. Okay, non-homologous is the single nucleotide bases. This is the difference. For homologous there is the duplex, and non-homologous is a single. So here is simple, at the damaged portion, the DNA damage portion will be detected and will be ligated using the ligation technique. This is the simple thing, and quick and dirty. Dirty means in the non-homologous end joining, there are some dirty molecules like the introns. Intron means the junk DNA. Those are the non-functional DNA. So that's why this is the dirty portion. So this is not so important for the genome reconstruction, because the dirty portion and the non-homologous chromosome contains maximum number of introns. So there are some junk DNA or introns. So there is no much importance to repair the non-homologous eventually. So this is the all about non-homologous and homologous recombination of DNA repair. Hope you understand. Thank you for watching.